Hello everyone, welcome to the Phenomics webinar. Very pleased to introduce today Katy Rainey. She will talk about managing UAS imagery for developmentally driven decision making and genetics analysis. But let me briefly also explain you some background on the webinar series and why we do it and who is organizing the webinar. So the basic idea is to develop an exchange and provide an exchange platform for the community that will to talk about plant phenotyping and about many different topics related to plant phenotyping. We specifically seek contributions from all plant phenotyping related work fields across all career stages. So please feel free also to submit an abstract. There is a platform where you can submit an abstract. We do those webinar series in two different formats, as a single talk, but also as thematic sessions, where we have multiple talks around a specific topic. The webinar series takes place every second Friday at 2 p.m. Central European time. So who is the organizer? So the organizers are, it's IPPN. IPPN, it's an association that aims at integrating and at the plant phenotyping activities across the globe. There is a very vivid structure with the IPPN office, board, many members focusing on many different topics, with the major goal to integrate the fragmented activities around phenotyping, enable and facilitate exchange, like with this webinar, address technology development, implementation, as well as advise different stakeholders on opportunities and limitations linked to plant phenotyping. Then within Europe, we have a project called European Plant Phenotyping Network, which is open to the community. And this project provides a transnational access based on application. So access to plant phenotyping facilities. Currently, there are 31 facilities available for access. So you can, it's based on a very simple application. You can apply, and once you are accepted, the full costs are covered, including travels, and 20% of the access includes people from outside of Europe. So it's open to any applications from outside of Europe, and the calls are every 6 months. Actually, we have right now the very last call. The deadline is 26th of October. And we are looking for your applications. And looking at the previous results, we have currently 33 different countries that are involved, 124 projects that are ongoing. There were so far 70 publications resulting, actually almost 80 right now, resulting from this experience. And it's actually a pretty nice way to enable experiments in different platforms. And additionally, emphasis aims at long-term sustainable continuation of EPPN 2020 to develop European organization that will provide access as well as other services related to plant phenotyping. So basically develop a pan-European infrastructure, provide access to those facilities and enable genotyping of performance in diverse environments and show knowledge, experience, technology. We are focused on 5 different infrastructure categories. You find some more details when you go to the website. There are some videos, very different, some interesting videos, and you can follow it on different media. And we recently also updated the GDPR, so please renew your newsletter subscription and any other subscriptions and be informed. Moving now on to the day, what we do today. So we are very pleased to introduce Katy Rainey. So Katy is an associate professor of soybean genetics and breeding in Purdue, and CTO of Progeny Drone Inc. So Katy works with unmanned aerial systems and is doing a lot of RGB imagery of soybean field trials for a while. She's also surveyed on multiple locations with RGB and multispectral imaging, completing over quite a substantial number of flights. Receiving a huge amount of traits or to quantifying canopy structure, canopy properties. And as I can see, you will also add thermal camera and thermal imagery into the portfolio. And as such, we are looking forward to Katy, to your presentations and to your presentation, and the stage is yours.
Okay,
And maybe sorry once more for the participants at the webinar. Please put your questions into the chat. We will address these questions at the end, and you can do it throughout the presentations at any time. So sorry, Katy, for interrupting. Now the stage is completely yours.
Okay, I'm just gonna try to figure out how I can switch back to sharing my screen. Okay, I think now you can see it, right? All right, can everyone see my slides?
Yep, perfect.
Okay.
Yep.
Yeah.
All right, well, thank you to Roland and Jennifer and Philip for inviting me to talk, or the IPPN. And so I was approached to talk about with an industry perspective, but also as an academic or a scholar, you know, without doing like a pure sales pitch. So I'm giving a talk that is similar to something I gave to Genomes to Field at the Genomes to Field workshop at the Phenomics conference in February, as well as some of my scholarly outputs and publications that I've given at a few at the Soybean Breeders Workshop. So anyway, just to begin, I want to acknowledge that all of my phenomics work is in collaboration with Dr. Keith Turkauer in Ag and Biological Engineering at Purdue. He's a hydrologist and has set up our data management pipeline, and we have a really good synergy in terms of he uses our yield data and other phenotypes for yield models, and that he uses in hydrological models, et cetera. And then, so we collaborate, we've been collaborating since about 2014. And this is an older picture of him with one of the kind of DIY type drones that people used to use before we have all the cheaper platforms now. And then the picture on the right is of my student who recently graduated. Her name's Fabiana Moreira. She now works at Bayer, but just acknowledging that all the things I'm going to show have student contributions for the most part. And then in the green at the bottom are some definitions. I won't talk about them. They're just there in case people need them. Okay, let me make sure I'm in PowerPoint. There we go. So I'm also presenting sort of on behalf of my company, which is Progeny Drone, but our software is called Plot Phoenix, and it employs Anthony Hurst full time. He was a student that worked with Keith and I, and then developed the plot clipping platform or software as part of his PhD, working with my data. And then recently, Edwin Rydell joined us as the business development manager. So my goal with this talk is just to present, my approach to field-based phenomics as enabled by the Phoenix software and some other contributions. So a little bit of background on me. I'm a soybean breeder, and I have a very typical soybean breeding pipeline here at Purdue. This is an illustration of what we had in 2019, which is a typical, our pipeline tends to remain consistent from year to year. So we have about 14,000 plots that we evaluate at, 3 or 4 locations in Indiana, and divided into things like the plant or progeny rows, and then, moving forward into preliminary and advanced yield trials as we do our selections, primarily based on yield, but also using the UAS data. And just a plug for the fact that I think breeding programs are good places or good formats for phenomics, in that we're evaluating a lot of low yield material. And the reason why this figure has that sort of inverted pyramid shape is because we are winnowing out we're throwing out the trash in the process of evaluation of material. And so you do end up collecting data on plant types, that have a range of phenotypic variants, which I think is important for phenomics. And I'll talk about that in a minute. So in my worldview, I guess the goals of field-based phenomics for a crop scientist or a breeder or an agronomist is can be primarily for selection or genetics. And I have the main goals asterisk. So people want to either predict yield or genetic yield potential. And this might be using genetically correlated secondary traits, or just anything that's helping to adjust your yield data to represent something true from a plot-based format. For genetics, we might want to conduct some kind of growth analyses or find genes that control how the crop is growing, whether that's in response to stress or pathogens, et cetera. Or we might be looking to identify a new trait or new phenotypes for a trait, or both, and then understand them genetically before perhaps applying them for selection. And then there are kind of two flavors of phenomics, I think, in the field right now that would apply to both of those overarching goals, which is to replace ground-based phenotyping methods. So you can convert visual scores into quantitative phenotypes to obtain better precision. And you've got, you can use a computer vision approach on images, for example, or data, or you can, do a more traditional growth analysis type approach with data collected, in a field-based phenomics experiment. So I think it's important as scientists, I advocate that when designing field-based phenomics experiments or trying to apply phenomics to something like a breeding pipeline, that scientists define their or your minimal spatial and temporal resolution, that the spatial and temporal resolution should be defined by the scientists, and it's definitely a moving target. I also advocate that there's a lot of biology and genetics to be done on growth analyses of phenotypes from RGB images. And so I urge people to recognize data snobbery and to really explore what can be done with RGB right now. And so my motto that was kind of presented before this began is do genetics now. So I really advocate that using any kind of sensor technology that gets us a lot of data from multiple environments so that we can apply genetics. And I really believe that that's where there's a gap right now, is in genetic analyses of longitudinal traits. And I'll get to that in a minute. And then it's important to collect images and other data when it's developmentally relevant. And that makes it hard to standardize data collection platforms or data collection protocols. For example, in Nebraska, it could be too windy by noon. In Indiana, it could be, too cloudy on certain days. You can have different soil types, et cetera. And so it's more important to focus on the developmental windows that we're trying to achieve rather than which platform that we use. And this is illustrated in this plot of biomass from my student, where you can see that the spatial and temporal resolution that might be required to predict biomass across these days after planting is really variable. Early on in the season, you might be wanting to get extremely high spatial resolution and temporal resolution to describe the beginning of that curve. And then later on, you're going to maybe, depending on how the season's going, you can adjust your goals in that regard. And then on the far right is an example of how stand counts require, extremely high spatial resolution, and you could have a window to collect, an ideal window for collecting stand count imagery of just a day or two. And that's why I think it is also difficult to have these traveling phenomics teams as a paradigm. I think that crop scientists need to be enabled to operate platforms and decide to collect data when they think it's important. So I was saying there's a lot of biology and genetics to be done on growth analyses from phenotypes of RGB imagery, and this is just an example of that. I have some experiments going on where we're using different dosages of various herbicides on soybean to simulate stress. It is a stress, and we're using herbicides for two reasons. One is I feel like it's an easier stress to manage than pathogens. There's a lot more complexity in inducing a disease, or even inducing an abiotic stress like drought stress. And so we're using herbicides, plus herbicide tolerance is just an important trait in soybean. And so you can see, like, here are three on the left are 3 dosages of glyphosate on glyphosate susceptible soybeans, and then there's the canopy coverage and greenness scores across time, and there's a lot of information there that can describe the stress, and that could be, so you could get similar curves for all kinds of biotic and abiotic stressors. And you know, and also I see people who are considering taking up phenomics that maybe too focused on measuring the thing that they are typically scoring visually, like so currently on the left, you would be describing the current method to describe that stress from the herbicide would be with a ground-based visual score. And you know, we're working to replace those, as I said, with something more precise and quantitative. And that can go for anything like the, you know, measures of herbivory, looper damage, anything like that. And I encourage people to think about not replacing that visual score, but really what is the stress that your favorite, what is your favorite, what is the impact of your favorite stress on the growth and the canopy coverage and color, et cetera, and rethink what you're trying to measure and what that's enabled by particularly the UAS platforms. Another illustration of the need to have phenotyping protocols that are responsive and specific to a season and a location is illustrated here. This is the distribution of canopy coverage in 2 different years in one location. You can see the vastly different growth from one year to the next. And over on the right is an example. This is the soybean should not come up to your shoulders, but in some years they will grow that way. So it also emphasizes, so you know, what is your longitudinal trait data or your phenomic data mean in that scenario? And also it illustrates something I didn't say earlier, which is I really think for field-based phenomics and at least the row crops that, you know, UAS imagery is the way to go in terms of getting enough data for doing genetics, because you see that your canopy turns into an ocean of green pretty quickly, and it's hard to do anything under the canopy. This is an example of a very standard type of metadata description that happens in my lab these days, which is to plot out by location or experiment, environment, however you want to call it. Like here we have 2 years and 2 different locations on the left, and then moving across, we're representing days after planting, and then both our flight and ground sampling dates are graphed as well relative to phenology. So R1, R5, R7, and R8 are phenological time points or events in soybeans that are important for interpreting your longitudinal data. So I have my students, this is just like doing a histogram, for example, of your quantitative trait data. You might want to also just look at a plot like this before even launching into your analyses. And it also shows how, you know, phenology is different every year. And so, you know, it's hard to prescribe you should, you know, collect your data at, let's say, 40 days after planting, when you have a lot of variability from year to year and how the plants are growing. So I liked, in terms of approach to managing data or designing experiments, I like to dispel the notion that, you know, a lot of times people will call drone imagery data, and it really isn't the data. It's an intermediate form of the data that has a lot of post-processing that needs to be done in order to make use of it. I'm sure everyone on the line is familiar with that. And I also, I recognizing that I just, or I would like to acknowledge that I just told everyone to recognize data snobbery, and now I'm going to impose data snobbery by really strongly advocating to take your raw imagery and extract plot clips from it, which is what is enabled by the software that my company provides. But just to be neutral, I'm providing in the top right corner a citation from earlier or late last year that has a plot clipping protocol in it that can be used in Python. I think from orthomosaics or from certain software like I think Agisoft or Pix4D. You can take those kind of outputs and then clip plots with that pipeline. So you don't have to pay for our software in order to obtain plot clips, which is a sort of moniker that our company has trademarked, the plot clips. So so I really advocate, the orthomosaics are pretty pictures. They aren't designed for data collection because they're representing an average of all the hundreds and thousands of images you may collect on a given flight date. And it's better to, it's preferable, I think, to get these plot clips and then derive your phenotypes from them. So this is an example of processed plot clips of uniform pixel dimensions that come out of the Phoenix software, and each plot is labeled with the sort of experimental metadata like the date, the camera, the location, the experiment, and the range and row coordinates of the individual plot. So you end up because of you always want to achieve as good as in terms of your spatial temporal resolution. An element of the spatial resolution is your overlap in images, which should always be as high as you can possibly afford, given some matrix of priorities, and which will give you replicate plot clips anywhere from 2 or 3 to 20 images of each plot, just depending on wind gusts and your platform, etc. And so this is illustrating along the bottom, there's 6 images of the same plot and that from different perspectives that you can then quantify phenotypes from those images, and you're going to get some advantages from that. The plot clips are going to preserve your raw image colors and sharpness, particularly if you shut off your default camera settings on an RGB camera that may be programmed in there in order to achieve pretty pictures, and you want to maybe turn some of those off. You could talk to our CEO, Anthony Hurst, about that if you have questions. Your plot clips will trace to an original raw photo, and this would be plot clips from the Phoenix software or from another pipeline. Usually you can trace them to the original raw photo, and then that will give you information on where the UAS was when the photo was taken. That's the kind of drone-based metadata. Your plot clips can be filtered, discarded, and subsetted for quality control, like you can QC for blurriness or the location of a plot within the photo, with the higher quality plot clips coming from photos that are where the plot's sort of in the middle or on nadir perspective to the plot. You can also correct for glare, etc. Plot clips can be batch processed because they're labeled the way they are, and they can give you a standard deviation of your phenotype, which is going to give you some statistical analyses options and quality control, etc. The other thing that's nice is that the plot clipping approach, at least as Phoenix enables, avoids high performance computing and cloud computing. So with the Phoenix software, you can derive these plot clips without any kind of uploading your data to the cloud and waiting for it to be processed. And so for most types of experiments, you can get these plot clips and the phenotypes quantified from them within, an hour or two. You can even do it before you get back in your truck and leave the field, and go through certain steps, at least in the pipeline, to ensure that you have the data you want before you leave the field, okay? Because the orthomosaic pretty pictures are very computationally intensive to generate, as well as sort of unwieldy data products to manage. So the Plot Phoenix software will output these plot clips without RTK GPS. There's no need to have ground control points, terrain models, polygons and shape files, orthomosaics, cloud computing, or high performance computing, as I said. And it's automated and can be done at the field's edge, which has the advantages I just mentioned of allowing you to phenotype and QC your phenotypes and imagery at the site and make decisions. So here's another output from Phoenix that would be the replicated phenotypes that are quantified from the plot clip images. So you can see location in this sort of spreadsheet that it puts out, location, experiment, any kind of metadata you might want to customize for your drone imagery. And then you're seeing a median, reference, crop cover, and green-red ratio. We have more metrics than this now. So, you know, you can make decisions in the field with those data pretty quickly, or you can do your flight in the morning, maybe when it's not windy, come in and have lunch and process your imagery, and then go back out and say subsample, you know, based on these phenotypes, subsample your field. This is an example of another output from Phoenix, which is a heat map with the sort of real-time UAS phenotypes. So you can generate this map, go all the way through to generating this map at the edge of the field if you wanted to, and you can go out and look at the plots that are maybe different, or again, come up with a subsampling strategy based on these data. The reason why you don't need to use the RTK, GPS, et cetera, is that the thing that Phoenix is doing is using the range row coordinates of the plots and some other data and some other metadata about your experiment that you input into the software. And you know, I think it's worth noting that for the most part in field-based phenomics in agriculture, your plots are going to be, I mean, your experiment is in a plot. It couldn't be a simpler thing to look at in terms of being in a grid that's very rational, right? So, and the range row coordinates are usually needed for any kind of GPS planters that you might be using to plant your plots. And so it just makes sense to go ahead and use that information. When in Phoenix, I tend to advocate to always process your borders. It's just easier to include your borders throughout your image analysis, and then just strip out the phenotypes when you don't need them later on. And also, we do recommend at least flagging certain areas of the field so that you can quickly verify that the image processing was done correctly. Because even though I just said we have this nice uniform grid of plots, there's always, you know, there can often be issues with planting errors, et cetera, and dog legs as the tractor turns around that can make, it can cause some issues, though there are a lot of features of Phoenix designed to adjust for all of those issues that you may have encountered if you tried to use shape files and things like that. So, and if you want to know the nuts and bolts of how to ideally set up an experiment in Phoenix, it would be, you know, you should set up an appointment to talk to Anthony or Edwin. So now I would like to move into giving some examples of research that we've done with this pipeline. This is a very busy slide, partly because this is showing some outputs that have been around for a while for us. We published a paper on this in 2017 based on data from, you know, 5 or 6, 7 years ago. But our first phenotype was just canopy coverage from plot clips, which, you know, % canopy pixels quantified over the soybean nested association mapping population that we were collaborating with a whole team to feed it, you know, with the main goal of mapping yield genes. And then we published a paper, my former student and some other folks, looking at the genetic architecture and quantitative properties of canopy coverage. And we published at the very top this Manhattan plot over time, where the p-value is rather than being the height of the hit, is the heat map of color. And so we were, you know, this is describing the genetic architecture of, you know, what genes may be associated with this canopy coverage development throughout development. And that just was really eye-opening for me in terms of how I approach phenomics now, that whole process of, and so it was a very simple phenotype, and we went all the way through to some advanced genetic analyses pretty quickly from, you know, 3 or 4 locations or environments of data. So we found just from data just from 3 locations within Indiana, 3 environments, as you can see on the far right of this middle table, we found QTL, you know, one big QTL that affected yield and maturity, and the growth of the canopy cover, that this QTL was also identified from 17 environments of yield data. So this is illustrating, how the, again, there's a lot of genetics to be done with RGB. Like I think trying to map growth with RGB images is going to find a lot of genes that you could identify through more labor-intensive or painful, expensive phenotyping methodology. And then what's happened since we published this is that a couple of other studies, which moving down towards the bottom of the slide, have found the same QTL that we did using completely different germplasm. So it's really nice. That was a huge validation to me that that what we are seeing is real. And I think it's important to combine genetics and phenomics, at least if you're thinking about crop science, so that you can really think that your phenotype is something that has a genetic basis. So it's exciting to see other people find the same QTL. And I've had other people, the studies haven't been published, suggest that the QTL we found is maybe associated with branch angle. So it's kind of cool to think that, again, you can find the same QTL through multiple phenotyping methods that are more or less painful. And then our data were public, and another group found that the highest correlation between canopy coverage and yield happened about 30 days after planting in soybean. And we found that the genetic correlation between yield and canopy coverage was quite high, which led us to this next experiment that's been published, which was to look at selecting in our project. I have a breeding pipeline, so we wanted to use the data in their pipeline to select our progeny rows. And the Venn diagram represents 3 different selection categories that using yield, canopy coverage data from drones, and then yield with that canopy coverage as an adjustment. And what you want to see in a Venn diagram of selection categories is some overlap, but not total overlap, right? The traits are selecting the same lines. It's a good sign. And so we repeated this over 2 cycles in our breeding pipeline. So that's why I'm going to be representing 2 years, but it's different germplasm, because that's what happens in a breeding program, right? You advance your material. So we applied these selection categories, and then we divided our lines into just early and late maturing. That's a soybean thing, and tested yield over 2 years in our preliminary yield trials. And these figures show the distribution of the 3 selection categories and their significant, whether or not there's significant differences in yield. And the result we saw in 2016 is that there was no significant difference in yield between lines selected with canopy coverage, yield, or yield adjusted for canopy coverage. As a general summary, which is very significant, because you can make your selections based on canopy coverage very cheaply and by the middle of July, as opposed to going all the way through to harvesting yield, the plots and measuring yield in September, October. And then that result didn't necessarily repeat in the second year. We have subsequent data across multiple breeding programs that my lab is still needing to analyze. But you can see that in 2017, the yields, the selection category of yield did outperform the others. But in plant breeding, it doesn't really matter what a distribution of material does. It doesn't matter about the performance of a distribution of material. That what's really significant is what selection categories pick the top ranking lines, which is what's summarized here. So you have early and late advanced yield trials from 2017 on the left and 2018 on the right. And we're showing the selection categories for the top 10 ranking lines in those trials. And anything in red would not have been selected without drone data. And so that's just really emphasizing that it can be useful in certain years at least. And then that the yield adjusted for canopy coverage selection category there at the bottom, you can see it's selected more top ranking lines than yield alone. So that yield given adjusted for canopy coverage is, I think, adjusting for non-genetic effects largely, like variation in plot size, row length, gaps, etc., because soybean progeny row yield data are really low quality. And I know at Corteva, they use this yield adjusted canopy coverage metric for all yield trials for soybean, not just progeny rows. So then my lab moved on to wanting to predict like temporal soybean biomass or longitudinal biomass traits, with the motivation that biomass is an important component of many physiological efficiencies like water use efficiency or nutrient use efficiency, and that the sort of decades of yield studies in soybean had demonstrated that increases in biomass are what are driving yield increases over time as a result of plant breeding. So and I'm probably running out of time, so I'll skip this. But the main kind of takeaway for our design of our experiment is that we wanted to collect our phenomic. We designed our experiment to be sort of a compromise, I think, in terms of the cost and effort. How could we explore our ability to phenotype biomass and then traits that related to it without spending a lot of money, and get some genetic data to understand the traits and where they could be applied in the breeding pipeline. And the other thing is that when you want to look at genetic correlation to yield of a trait, which to me is the most important thing as a breeder, I want to know what's predicting yield. You need to be measuring yield on low yielding material, which is not typical across all of agronomy. And so I think that's one thing to note. So we had a lot of phenotypic variation for yield in our biomass experiments. And then also it's important to think about whether or not the material that you're phenotyping in Phenomics has been selected for yield. Even like association panels of land races and things, those lines have been selected for yield so that whatever phenotypic variance is displayed might not be what's predicting yield. And so I think it's best when designing like a phenomics experiment to maybe subsample a magic population or a NAM population. So you've already seen the one figure of the phenology, but just pointing out that I think I have these out of order. No, I just skipped the important slide. So we put together a subset of soy NAM for our experiment, and we controlled for maturity because phenology is so important, based on all the soy NAM data that was available, and then just randomly selected lines and represented a pretty large phenotypic variance for yield, and put that out into experiments that were 6 to 8 row plots so that we could collect ground reference biomass data over time from bordered plots, as well as having yield from that same bordered yield data from plots. So we flew and ground sampled pretty frequently, and we imaged this experiment with multispectral and RGB cameras, and this is where collaboration with Keith's lab, his students came up with a plot clipping pipeline for multispectral imagery that's been published. And so it's possible to apply plot clips to multispectral imagery, though it's not currently a feature of the Plot Phoenix software. And my student was able to pretty reliably predict biomass within location, couldn't do it across locations, using machine learning applied to all these vegetation indices, as well as the RGB canopy coverage phenotypes. And it is projected out over sampling dates because we had more flight dates than we had ground reference dates. And we're trying to currently get this study published. And moving on to the results, this is showing the soybean biomass from this panel of about 400 soy NAM lines summarized over the 3 environments, and about 60 days of the growing season. And this is coming from over 15,000 biomass phenotypes estimated from the UAS imagery for each flight day, using that machine learning, a lasso prediction. And you can see we had good yield, or good range of yield, and a high, or typically high average yield in this experiment, with a high heritability for yield. So it's good quality data, good quality plots. And then to make genetic inference of these phenotypes, we applied random regression models. This figure is coming from a paper that we published this year. It's a review paper on your options for genetic analyses of longitudinal data, which again, I think is a gap right now. And we actually applied models in collaboration with quantitative geneticists in animal breeding that were developed primarily in dairy cows to describe lactation curves in dairy cows. So with the output, not a single time point analysis, but finding genes that control the shape of the growth curve, not the phenotype at a given point in time. And the thing that random regression analyses allow you to do is to interpolate what your quantitative properties are on any given day, or whatever description of development or time that you want to put on something, rather than just limiting it to the time points that your data were collected. So here is an estimate of narrow sense heritability over those 60 days of the season for biomass. You can see it's low to moderate and really changing throughout the season. And we also looked at our sort of genomic prediction accuracy of for biomass over time, and which declines as the season progresses. And I think one thing that will really help with this is adding height into our estimation methods, which we're currently working on. My student then can perform UAS analyses using random regressions, and found SNPs that she could sort of classify or bin into their period of effect as either long, mid, short, or intermittent, as shown on the far right. So on the left side of this figure is the individual SNPs with their location in the genome. On the bottom x-axis are the days after planting, and the far right's describing these categories. So you can see, and the heat map is the SNP effect, whether it's negative or positive, and how strong it is. You can see there are SNPs that are affecting biomass production in soybean for 30+ days, some that are doing it for maybe 10 to 30 days, and some that are less than 10 days, and some that are kind of coming and going in terms of their, some of the SNPs in terms of their effect. How we use these data in breeding, I'm not sure. I'm more interested in the genomic prediction of the biomass and incorporating that into genomic yield predictions, which is what my lab's currently working on. The other thing that we're working on now is improving our biomass phenotypes using 3D modeling. My former postdoc just went on to, she has this sort of a staff position now at Purdue to be doing this across multiple crops. This is a 3D model of a soybean field from the, overlapping images from the drone. And here's some phenotypes she's derived from that. We have papers published or coming out about this, showing height variability within the plot. These are themselves phenotypes that can be described. And then just moving on, and I have an AFRI grant to incorporate this approach with our multispectral and RGB-based canopy coverage phenotypes to try to improve our biomass prediction accuracy, and then test it across like multiple germplasm from multiple public breeding programs, and look at using biomass in a genomic prediction model for soybean progeny rows. That's one of the other things we're currently working on. The Plot Phoenix is about to release a sort of Plot Phoenix 2.0 version of the software that my CEO likes to tell me is going to be twice as fast and twice as easy. But the real thing that I'm so excited about is that we're adding plot height. So we have the ability to generate 3D point clouds by plot, not over the whole field, but by plot, which really cuts down on the computational load, et cetera. And we're going to be outputting both a height estimate for plots as well as the point cloud data that can be really mined for all kinds of things. So I'm so excited to apply this metric and to improve our biomass phenotypes and all kinds of other things. And you can see we have a new sort of improved user interface coming along, as well as some improved. It's really challenging, like I said, when you have errors in your field and things that aren't quite square to adjust, to clip the plots for the range row coordinates. And so there's some improved functionality there coming out from the Plot Phoenix software. So I'll be looking for that. And with that, I think I'll see if we have any questions.
Yeah, thanks a lot, Katy. It's a great introduction of the field phenotyping and use on drones for breeding and for assessing plant traits. So there are some questions. So let me start with a question from Darmodar Poidal. So he asked if is it possible to identify and locate off-type like mutants or non-hybrid plants among hybrid plants in a large plot by using image analysis?
Well, I don't think the plot Phoenix software doesn't have that metric that I'm aware of, but you could probably, if you had sufficient resolution in terms of your, flying relatively close to the high overlap, low altitude close to your canopy, if you had something that was different in height, different in color, etc., you could probably take the plot clips. So the plot Phoenix software will give you the plot clips in different formats actually, and you could apply some sort of machine learning algorithm or something to that perhaps to flag your plots for evaluation for off types. Yeah, no, so we haven't tried doing that yet. I think getting the height metric and validating it and exploring its capabilities and the sort of coming up with best practices for collecting imagery to get height-based metrics is going to really be interesting, and we can explore how to use that to identify off types.
Mm-hmm.
But right now, I don't think that we don't have the capability to track like individual plants. So I think you could use the imagery to do that if you came up with your own, the plot clips if you came up with your own.
Yep. There is also a question by Darmodar. Can you comment on the use of this technology for the detection of pathogens in a plot?
Well, I think that it's a great question. Again, it's about looking at canopy cover and greenness and those changes over time. So don't think about, can it detect frog eye leaf spot or can it detect tar spot, but what are those pathogens doing in terms of the growth of my crop, and then looking for, perhaps coming up with a regression of canopy coverage, greenness, and those changes over time that may be predictive of that stress. So yeah, I think I think there's a gap right now, and I'm talking to people across the globe, I guess, who care about plant phenotyping. What I would like to see is on a crop by crop basis, white papers written about the ranking stressors in terms, or prioritizing them in terms of what's their economic impact? Do we have the ability to control them if we detect them early? Are they likely to be detectable given RGB or hyperspec or thermal, et cetera, and kind of describe the opportunities? And then, so yeah, I think there are probably some pathogens that'll be detectable and some that are not, just depending on, how they manifest. For example, sudden death syndrome in soybean is largely underground, so I'm not sure what we can do with RGB imagery, but it does produce a toxin and some other signatures that maybe the hyperspec could pick up. So yeah, I think there's unique opportunities that the communities of pathologists and engineers, et cetera, need to come together and describe.
Yes, thanks. So there is a question by Jess Alvarino. So I'm gonna read this question, which is actually, so it starts with, I've become a fan of yours. Hi, Katy. I've become a fan of yours because I made the critique paper as my subject requirement in your article, improving the efficiency of soybean breeding with your throughput canopy phenotyping. It's awesome. So this is the question. How do you decide for the right altitude to acquire canopy images? Is it fixed?
No.
And do you, sorry, the second part of the questions. And do you consider the time of the flight and maybe cloud cover during image taking?
Yeah. So again, I think it's hard to globally prescribe a phenotyping protocol because every platform, every site, every crop is different. However, if you're trying to do this with your specific platform information, et cetera, you should definitely reach out to the Plot Phoenix team and they can help you troubleshoot, like Anthony and Edwin. plotphoenix.com. They can help you troubleshoot your specific hardware. But I will say that canopy coverage, at least in soybean for selection, does not have to be high resolution spatially or temporally. So, 4 centimeter per pixel resolution on that should be fine. And, anytime within a 10-day window would be fine as well for the most part, though we do know earlier in the season is more predictive of yield. And when you talk about early season, you're going to have smaller plants and rapid change. And so, you may want to, at least as you're starting your process of applying these phenotypes to err on the side of having more data than less, and then you can maybe adjust as time goes on. In terms of clouds, I'm not that worried about clouds. I mean, obviously you don't want it to be really cloudy, but like a canopy greenness, right? That is not a calibrated metric, but does it really matter? Okay, we don't care how exactly green something is. We want to know how green they are relative to a check or relative to each other, and a little bit of cloud cover, et cetera. If that's affecting the robustness of your signal, then it's probably not a signal that at least a breeder should be using, right? So a little bit of clouds is fine. I'd rather have, there is no perfect time of, it's hard sometimes to get the perfect window to fly your drone. And so I think wind is much more of a problem than clouds.
Maybe a related question, soil wetness or moisture when it's, so the color obviously changes, the background color, and does it have an effect?
No, that in terms of your ability to detect canopy coverage or the operation of Flat Phoenix software.
Mm. And maybe yet another question related to the same issues. It would be on reusability of data. For instance, when you have those kind of different issues, and when you look at relative changes, can you still reuse the data in a different context and compare year-to-year data images when you know that there was obviously different background light, background whatever?
Right.
So
well,
how? Mm-hmm.
I think breeders in particular, but maybe crop scientists in general or plant scientists in a field context, you don't want to be using a trait or a signal that is that sensitive to variation in conditions. So particularly if you have controls, which we do have controls built in, obviously for yield or other traits into our experiments, why not have controls built in for color, where you can standardize things relative to a yellow soybean or a darker green soybean, or just, so yeah, I don't think, and then I think that's actually an area that could be explored, like how do you adjust color data from RGB imagery to make it good enough for making those kind of inferences without having to know the exact spectral wavelength or reflection in your field at that moment in time. As long as you're turning off those RGB camera settings that are adjusting for ambient lighting, like on the fly, which will give you pretty pictures, but maybe are causing too much adjustment, or you want to have your camera settings right, which is usually a matter of getting in and changing the default ones, which Anthony can advise you on.
Okay, thanks. There is another question by Canela Vieira. Is Plot Phoenix coming up with an automated maturity estimation feature soon? So,
yeah, so right, we want to replace those. No, I think you would want to apply the greenness index as published by Corteva a year or two ago yourself. And maybe someone could put that on GitHub or in R or something, how to do that calculation. No, I don't know that we have that as a goal, because we don't right now keep track of, Plot Phoenix will not keep track of your data over time for you. You can collect your imagery and label it to reflect when it was collected, all these things, but then it's going to be up to you to combine your data over time, which is, I think, what you're going to need to do for the, right, like the best estimate of maturity that we have right now in soybean is that inflection point of color. And so you're going to have to track your plot over time to achieve that.
Mm-hmm. Thanks. Question by JD. Are there other optical technologies or platforms that you would like to explore if you had the time and the resources?
Well, yeah, my lab is, like I said, we have multispectral sensors and then thermal, because I think that differences in canopy temperature during pod fill or grain filling are going to be really predictive of yield and a nice way to augment canopy structure information from RGB. Hyperspec, I'm not as, that seems too expensive and emerging as a technology that it's not going to allow me to do genetics, and even the thermal, maybe not. But, and I know there are challenges to thermal and getting that correctly, but Keith's group is really innovating with one of my thermal sensors. So I have hope that we'll be able to do it relatively well as in a plot clipping, scenario. That's what they're working on. So that's what I'm interested in. But yeah, no, really just very excited about 3D modeling. And once we get these point clouds by plot, which is really maybe going to be more accurate than over the whole field because of the elevation changes across the field, then yeah, just really exploring what you can do with that in terms of predicting, lodging or canopy roughness. Yeah.
Thanks a lot. Are there any further questions? There is. What are the computational needs for your software? Which is hardware requirements?
Yeah, we have it listed on the website or in one of our webinars. So if you go to the Plot Phoenix website, there's some really useful introductory videos as well as webinars about specific traits. But I will say that, Anthony, the CEO, loves to emphasize that you can do it on a laptop, and it would be about a $1,500 laptop right now. So you want a solid state drive and some other things. You need to have, decent RAM, et cetera, but it's definitely not in the high performance computing, range at all. Relatively decent laptop will work just fine. And it depends on your goal, right? Are you trying to QC your drone data before you leave the field? Maybe you want a gaming laptop and go, just to process things faster. So Anthony can advise you on that. But yeah, it's not that, and it works with any platform and any camera. That's one of the things that's unique about Phoenix is we're very platform agnostic. Now you can get, so I would say you could get started with Phoenix with couple $1,000s in hardware in terms of the camera, drone, and laptop, as long as you're not trying to cover hundreds of acres.
Yeah. Thanks. Maybe a question related to that. How many breeders are actually using drone-based technology in their breeding programs? Do you know any estimate? Or some background on this information. My impression is that breeders are still have a very conservative approach, and they trust their visual assessment. So this is still their gold standard kind of. So what's the impression? How easily do they take it up, the new technologies, drone-based imaging? What's the impression?
Well, I keep being told that at the private companies in the row crops that, half the traits or something are being replaced with UAS, but like soybean, corn, wheat. I think in the public sector and then you start talking about all kinds of crops beyond row crops, no, there isn't good adoption. I really hope that our software and my presentation today and my general message will help with that, because there is this prevailing thought that Phenomics is too hard. I can't do it. It's too expensive. I don't, I got a bunch of images, but now they're stuck on a cloud somewhere and I can't quantify anything from them, et cetera. And I just say it's not that hard and not that expensive to get into, right? And I think the crops, and so the main thing is maybe getting one of your technicians trained for the 107 certification if you're in the US. So, you have to have someone who can operate the drone, which that is has gotten so much easier in just the time that we've been doing things in the past 5 years. Drones have precipitously dropped in price. They've gotten much more automated and easier to use. You still have to be certified, which in the US is like a month of work and $150, like a month of a couple hours a week at about $150, and you can get your certification. And then your technician should be flying pretty frequently. And but then with Plot Phoenix and a decent, computer, you can process your imagery yourself and start looking at, and then there's whole approaches to, okay, a breeder, you're saying that one of the issues is breeders trust their ground-based visual estimates. So they're going to have to go through a process of comparing, which isn't that hard, right?
Yeah. Yeah. So there is also a comment from Edwin saying that we have customers now in vegetable breeding, which is so this is good to hear that customers pick it up and people pick it up for their breeding approaches, which is great.
Yeah, good.
Yeah. So any other questions? I think if there are any other questions, I think people can contact you directly. The email was so that there is a website. Your email was, sorry, I don't remember anymore, but
well,
it was somewhere on the website.
Yeah, I'll type it into the chat. I mean, if you want to email me at purdue.edu, or I have this. You can also do krainey at progenydrone.com. Either of those will get you in touch with me. So I'd love, so thanks for all the great questions and your attention. I'd love to hear from any of you about your thoughts.
So thanks a lot, Katy. As such, enjoy your Friday morning. For us, it's already Friday afternoon, so we'll enjoy the weekend.
Yep.
Thanks a lot.
Thank you,
Katy.
All right, bye.
That was great. Thanks all. Have a good weekend.
You too.
Bye-bye. Thank you.